Muito obrigado e boa tarde. I'm going to speak in English. So thank you very much. It's a pleasure to be here in São Paulo. This afternoon I'm going to give you an overview of what has been done in human clinical trials to date on the path towards an HIV cure. You've already heard a number of talks that have covered in considerable detail some of the biological mechanisms by which HIV maintains latency and some in vitro approaches to attempting to either sustain latency or to reverse latency. And you'll be hearing later from Mattia about additional approaches for latency reactivation. But I'll be talking now about what have we done so far in people with HIV infection and what needs to be done. And throughout my talk, I will summarize a series of trials that have been attempted and then complete each section with some challenges that remain and questions that need to be answered. These are my conflicts. I consult for a number of companies involved in this research area. So why are we searching for a cure? We know that HIV therapy is incredibly effective. We've reached a point now where in European and United States and in many South American countries, if people get on treatment soon enough, they can live essentially normal lifespans. And in Southern Africa, we've seen an increase of a decade in life expectancy since the introduction of antiretroviral therapy. Well, among the problems with lifelong treatment are the need to continue taking it every day. That means that there may be side effects and toxicities that are not apparent in the first 3 or 5 years of treatment, but become evident after 15 or 20 years of treatment. In addition, there's the burden of long-term adherence. It's not taking a pill for 6 months or a pill for 2 years, but a pill for the rest of your life. The cost of continuing to provide that therapy is staggering, and there is serious concern about whether the ability to provide antiretroviral therapy on a global scale is really sustainable. We also know that the persistence of HIV and the persistence of HIV antigen and the production of virus particles, even on suppressive therapy, leads to continued inappropriate immune activation that may play a role in accelerating the occurrence of cardiovascular, central nervous system, and other end organ damage. If anybody stops treatment and has viral rebound, they may then transmit virus to their partners. And there's the ongoing stigma of HIV infection, and one of the main reasons that people living with HIV are interested in participating in studies of HIV cure is the hope that if they can be cured or enter a sustained remission, that they will be able to rid themselves of this stigma. So just very quickly, and you've already seen the detailed biology, so I won't spend much time on this, but as a cartoon of why is HIV persistence such a problem, most T cells are happy T cells, and then they encounter the virus and they become unhappy T cells, they get infected, they make more virus, and they die. And that would be the end of the story if it weren't that sometimes these cells go back to sleep and then they have an integrated HIV provirus which will persist with that cell for as long as the cell lives. And if that cell undergoes mitotic expansion through proliferative signals, we now know that these cells can maintain the state of latency while increasing in number. Occasionally, one of these cells will encounter its cognate antigen or some other cytokine, some other stimulatory cytokine that reactivates the cell and leads to production of more virus and kills the cell. But as long as these cells don't get stimulated but merely proliferate, there will be persistence of the virus, and antiretroviral therapy is insufficient to eradicate it. So let me give you some definitions because they're important in thinking about how we approach this problem clinically. By the reservoir, what I mean are cells that harbor replication-competent proviruses that could rekindle HIV replication or be responsible for transmission. So the vast majority of proviruses, which are defective, don't really constitute a reservoir. They're simply garbage. Unfortunately, when we measure, many of the ways we have of measuring provirus don't distinguish between the garbage and the small fraction of important proviruses, which is a major challenge in assessing the impact of any of our interventions. By a latently infected cell, I mean cells that harbor such replication-competent proviruses but in which HIV is transcriptionally silent and viral proteins are not expressed. And you already heard from the earlier talks why, if a cell is not making any viral proteins, it's essentially traveling incognito to the immune system because it can't be recognized as an infected cell and therefore can't be targeted for destruction. So by cure, I mean the elimination of all cells carrying replication-competent proviruses that could lead to recrudescent viremia and disease progression or viral transmission. And by remission, I mean some period of time, and nobody has yet said what that period of time should be, during which antiretroviral therapy would not be needed in order to maintain a viremia-free state without risk of disease progression and without the risk of transmission or the occurrence of non-AIDS events. Now that might be a year, 5 years, or 10 years, but so it wouldn't necessarily be a permanent state of cure, because there would still be harbored someplace these replication-competent proviruses, but at least there would be an extension of the time during which treatment was not needed. So by way of background, and you've already heard some of this information in the earlier talks, there's been substantial progress that's been made in understanding the mechanisms of latency, the anatomical and cellular nature of the latent reservoir, the genetic structure of proviral DNA, and the contribution of the clonal expansion of latently infected cells to the preservation and expansion of the reservoir. And there have been the development of therapeutic approaches in other disease areas that might be repurposed towards HIV cure, which has stimulated a lot of excitement and allowed us to begin embarking on pilot clinical trials with some of these agents. So there's a single example to date of a person who appears to have been cured of HIV, and that, of course, is Timothy Ray Brown, the so-called Berlin patient. Mr. Brown had long-standing HIV infection, was on suppressive antiretroviral therapy, and then developed acute myelogenous leukemia, went off his therapy in order to be treated for the leukemia, had the expected rebound in plasma viremia, and then his leukemia relapsed, and so he had to undergo a stem cell transplant. And he had the good fortune to have a hematologist who was quite forward-thinking and considered the possibility of identifying an HLA match donor who was homozygous for the CCR5 deletion. CCR5, of course, is an essential co-receptor for viruses that are CCR5 using, or R5 viruses, as we call them. And this being in Berlin, and where Northern Europeans have a 10% heterozygosity, and so 1% of Northern Europeans are homozygous for the deletion, it wasn't that hard to find a donor, and the stem cell transplantation was done, and from the time of the transplant, Mr. Brown remained free of HIV viremia despite having stopped his antiretroviral therapy. His leukemia actually relapsed, and remarkably, they were able to go back to the same donor and get additional stem cells from the same CCR5 deleted donor, do a second transplant, and this time the transplant was curative, so that the patient no longer had leukemia, and still is free of leukemia, but also no longer had HIV. And now this is a decade later, and after extensive assessments, some of which Steve Deeks, who's going to be here later in the week, has been involved in, there's really been no credible evidence of any persisting HIV in this patient. But this is an N of 1. And so far, although many other patients have received similar transplants, none have yet survived to be able to say that they are cured. There is at least one person still on antiretroviral therapy who received a similar transplant, but we don't know if that person is actually cured yet or not. Now, we had the opportunity in Boston, with the work of a former trainee of mine, Timothy Henrich, to study some patients who had undergone stem cell transplantation for lymphoma in the setting of HIV infection, who, in contrast to what was then the standard practice in the United States, continued on their antiretroviral therapy throughout the transplant period. So when they got these new uninfected stem cells, which transformed or matured into CD4 positive T cells, those cells were protected from becoming infected by antiretroviral therapy. And when we then went to study these patients, we could no longer, the patients became fully chimeric, that is, their entire hematopoietic system had been replaced by these HIV uninfected stem cells and now mature cells. We could no longer find any evidence of HIV in their circulating cells. They were on antiretroviral therapy, though, and we couldn't find in this particular patient who agreed to a rectal biopsy, we couldn't find evidence of any proviral DNA even in rectal mucosal CD4 cells. So after a lot of deliberation, we thought it might be appropriate to consider stopping treatment and see what would happen, because we could not say that these patients were cured. We had no test sensitive enough to say that the patients were actually cured, and the only test of cure is to stop therapy. So we stopped treatment and started monitoring every week, and what you see is, in contrast to what typically happens, and this is a scale in days, so here there's no proviral DNA, there's no plasma viremia, and typically we would expect by day 21 that virtually everybody would have rebounded. This patient actually went out beyond 200 days, and then after day 214 or so, came in, had a visit, had an undetectable virus, and then called 3 days later to say, you know, I'm not feeling that well. I have a fever, I have muscle aches and pains, I have a headache. And so we said, go right straight back to your doctor, get another virus load test, and here he was at 1 million copies. And then he went back on therapy and was able to resuppress. So what did this experience tell us? And I should have mentioned, these donors were wild-type donors. They had CCR5. So what we learned from this is that it's possible to substantially reduce the reservoir of latent HIV through stem cell transplantation. And because we know that in our patients who had previously undergone 2 cycles of autotransplantation and had gone through ablative chemotherapy in the past, that ablative chemotherapy by itself does not eradicate the reservoir. But the dramatic, and doesn't reduce the reservoir, but the dramatic reductions we saw in this case most likely were attributable to immunological mechanisms mediated through graft versus host reaction. But, and we also can say that reduction in the reservoir through this mechanism prolongs the time to virologic rebound. These patients had no measurable HIV-specific cellular immunity. They did have low levels of persisting antibody, but no CD4 or CD8 response to virus, because their circulating cells were now all HIV naive. And so it also appears that latency can be maintained in the absence of any antiviral specific cellular immune response. We can't say that you don't need antibody, because again, they were still seropositive, but no cellular immune response, and they could still maintain latency. This is true virologic latency. Nevertheless, virus can persist in rare cells that cannot be sampled by any clinically reasonable method, and those rare cells can reactivate and then lead to a recurrent infection. This led Alison Hill, who's a mathematical biologist or an evolutionary biologist now working at Harvard, to develop a model that asked how much of a reduction in the reservoir would we need in order to be able to say that it was likely that an individual patient would have a sustained remission or potentially a cure. So in our patients, we could measure a 3-log decline in their reservoir, and here I'm using the term very loosely because we were just measuring proviral DNA, and I already mentioned that most of that is not important. But if you reduce the reservoir by 3 logs, as shown in this line here, you only have about a 50% chance of going for about 2 years, or year and a half rather, before you rebound. In order to be able to get out to 3 decades, you need to have a 4-log decline, and then you still only have about a 35% chance of surviving 30 years without rebound. If you really want to be able to have lifelong remission, you need to be reducing the reservoir by more like 5 or 6 logs, and we simply can't measure that large a reduction because it's beyond the sensitivity of any of our assays, given the low frequency of infected cells that circulate in the plasma and the problems of doing serial tissue biopsies and knowing that you're sampling the right tissue in order to make these determinations. So with all the information that we've learned, what's now become the central dogma of HIV cure is that it's going to take some combination of viral reactivations through latency reactivation and some means of enhancing and harnessing the antiviral immune response, sometimes called kick and kill, although given the aggressive tendencies of our current leadership in the United States, I really prefer something less warlike, and maybe reactivation and elimination is a better way of thinking about this. And there are a number of different approaches, several of which have entered into pilot studies, and I'll show you some of the evidence for them. So first, to think about cell-based therapies. So one approach that has now been studied in several clinical trials is to attempt to sort of reproduce the Berlin patient experience, but by taking mature CD4 cells and genetically editing them so that you delete the CCR5 gene using either, in this case, a zinc finger nuclease or potentially Cas-based or Cas9-based therapies. And so the approach is to do a leukapheresis to deplete the monocytes and CD8 cells so that you have a CD4-enriched population, activate the cells, and then transduce them with either an adenoviral vector or a lentiviral vector that introduces the nuclease or the gene editing machinery of interest, then expand those cells and reinfuse them. And this was done by Carl June and Pablo Tebas in collaboration with the people from Sangamo. And what you can see is that they, in this case, after infusion, they interrupted treatment, and nearly all of the patients, well, everybody had a rebound, and nearly all the patients, the rebound continued until they restarted antiretroviral therapy, as shown in the dashed lines. But one participant actually spontaneously controlled viremia without having to add antiretroviral therapy. So this study was then repeated, and Jay Lalazari presented data where there was a substantial increase in CD4 cells with these transfused cells, but no real impact on the reservoir. And so what we've learned to date from these approaches, and this is all with mature T cells, there are ongoing studies with stem cells, but those have been limited so far to people who need stem cell transplantation, that these approaches are generally safe, that the genetically modified cells persist for quite some time, and I haven't shown you that data. And actually, when you stop therapy, the genetically edited cells actually are enriched, not surprisingly, because they're resistant to HIV infection. But it's been difficult to achieve consistently high proportions of gene knockout, and you need a double gene knockout because we're diploid, and many of the knockouts are just haploid. And it's unclear really how genetically modified cells are actually going to address the reservoir. It's one thing to say, I now have circulating cells that are resistant, but it doesn't get rid of the reservoir of cells. So this approach by itself wouldn't be sufficient to eradicate the reservoir. So where are we going with stem cell transplantation and other forms of cell-based therapy? Well, the Berlin patient experience really remains to be confirmed. We need to see whether this result can be replicated using genetically engineered autologous cells. We have to determine whether it's sufficient to introduce a population of cells that are resistant to the virus, or whether gene editing has to be coupled to other strategies to eradicate latently infected cells. And to see whether the phenomenon of graft versus host reaction can be harnessed or made more specific in order safely to attack cells harboring replication-competent provirus. Well, what about latency reversal? The early studies have all focused on histone deacetylase inhibitors, and you heard a bit about the basis of that approach earlier, and whether looking at vorinostat given as single doses in David Margolis's work, or as multiple doses in the work from Sharon Lewin, or whether looking at panobinostat or romidepsin. In each case, you get increases in cell-associated HIV RNA. So there's some modest level of increase in HIV transcription. In the case of panobinostat and romidepsin, there's been some evidence that there are actually increases in low-level plasma viremia, but these are really very tiny increases. But there's been no evidence at all of any effect on the reservoir as a whole. And as a group, these drugs, I'd have to say, have been relatively disappointing. Now, it's not surprising that the drugs aren't doing more than this little effect that we've seen so far, because if you look at what happens in vitro when you give maximum stimulation, either with the PMA and ionomycin or with CD3, CD28 beads to activate cells through immune signaling mechanisms, we know from the work of Yachiho and Bob Silicano and others that only a small fraction of the potentially infectious proviruses are reactivated at any time. And you can see here in this schematic from Yachiho's review paper that if the blue bowl represents the total pool of DNA detected by quantitative PCR directed against HIV GAG, then only a portion of that DNA actually represents intact provirus as determined by full-length sequencing, and those are shown in the red or purple bowls. And then the yellow bowl represents the tiny fraction that's actually stimulated by any round of in vitro stimulation. And if you took cells from this patient and stimulated them, and then added fresh medium and stimulated again, you'd get another small yellow bowl, and you could keep going. So you'd have to stimulate multiple times in order to actually reactivate all of the potentially infectious proviruses. And the fraction of cells that are reactivated to make infectious virus varies from patient to patient. So what have we learned with these very early efforts at latency reversal? Well, at current dosing regimens, the drugs we're using are really only modestly effective. And if maximum stimulation in vitro reactivates only a fraction of the intact proviruses, then reactivating the entire reservoir will really be a major challenge and would require multiple rounds of therapy. And it's highly unlikely that reactivation by itself is going to deplete the reservoir. We may need reactivation in order to be able to take advantage of certain immunological approaches, but reactivation itself is not going to get us where we want to be. So where are we going? Well, we have to see whether there are safer, more effective approaches to reactivating latent provirus, and there is at least one potentially promising candidate, a toll-like receptor 7 or TLR7 agonist that I'll say a little bit more about in a little while, that is now in phase 1 human trials and may be an alternative agent. We need to figure out how many cycles of reactivation will be necessary to induce the entire reservoir. Following on the talk we just heard, if there are uninducible cells that harbor intact proviruses, do we really care about them? Are they clinically significant? If we can't ever wake them up, why bother? Right? Maybe we should let sleeping dogs lie. And then if we're going to combine reactivation strategies with immunologic strategies, how do we sequence those? If you were going to use a therapeutic vaccine and a cytokine, do you do that first and then reactivate? Do you reactivate and then give the vaccine? It's going to take a lot of experimentation to sort that out. Well, what about immunological approaches such as checkpoint inhibitors, various cytokines, and other immune modulators? We know that chronic viral infections, and particularly HIV, lead to an exhausted phenotype where cells express high levels of PD-1, and it's possible to overcome this exhaustion by blocking the PD-1-PD-L1 axis. And this has been tried, at least tentatively, in one study that we in the AIDS Clinical Trials Group did. This study was led by Joe Iran, using an anti-PD-L1 antibody, so an antibody directed against the ligand rather than against PD-1 itself. In this case, this was intended to be a single dose escalation study where 8 participants at a time would get either the active antibody or a placebo, and we would start at the lowest dose and then continue to move up. Unfortunately, after we finished the lowest dose, or as we were finishing the lowest dose part of the study, some toxicity was observed in animals, and so both the FDA and the company decided that these studies shouldn't proceed in HIV-infected people anymore. This antibody has gone on to be approved for use in oncology, but no further studies are being done in people. But what did we learn? So 6 people got the active drug, and 2 of them showed significant increases in HIV GAG-specific CD8 responses, and this was interferon gamma release in response to HIV GAG peptides. In the 2 controls, we saw no such activity. And as in the case when these drugs are used in people, you see heterogeneity in the response. Not everybody responds to interruption of the PD-1, PD-L1 axis in terms of anti-tumor responses. Unfortunately, these drugs also carry with them the possibility for significant autoimmune toxicities, and this patient who had the strongest response also developed adrenal insufficiency and hypogonadism, we think because he developed inflammation of the hypophysis, the part of the brain that triggers the pituitary gland to secrete growth hormones that stimulate the endocrine organs, the adrenal gland or the testes, we know that there's about there's a several percent risk of developing hypothyroidism and a 0.5% chance of becoming permanently diabetic. And so these are not toys, and playing around with these in people carries significant risk. There are other antibodies, such as blockade, those that block alpha-4 beta-7, and in this case, a simianized version of this antibody, when given during acute SIV infection together with antiretroviral therapy, led to a state of remission where these monkeys continued to be SIV infected, but were able to control SIV viremia through mechanisms that are still not well understood. One of the reasons this is such an interesting result is that there is an approved drug for use in humans, vedolizumab, which is licensed or approved for the treatment of inflammatory bowel disease, ulcerative colitis, and Crohn's disease, and it's been very well tolerated in those patients. And so there's now an ongoing human trial at the NIH in Bethesda attempting to reproduce these results, and pending the results of that trial, there's enormous interest in moving forward in looking at the effect of this antibody in acutely infected HIV, people with acute HIV infection. Well, what have we learned from these very tentative approaches at using immunomodulatory therapy in attempting to reduce the reservoir? Well, there are clearly encouraging data from non-human primate studies, very limited studies in people so far. The toxicity profile of the checkpoint inhibitors continues to evolve in the oncology setting. And the regulatory agencies, like the US FDA, have shown appropriate caution in approaching trials of these agents in healthy HIV-infected people. The risk-benefit of these approaches still has to be determined because, as I said, there's considerable risk here. Now, we're about to embark on a study of cemiplimab, which is another PD-1 antibody, and the FDA is allowing us to do 2 doses of that antibody, but again, it will be starting at a very low dose and then escalating 6 participants at a time, and we hope to start that study sometime over the summer. Well, where are we going with this? There's obviously continued interest in exploring the roles of these therapies. It's going to be important to explore them in combination, but these combinations may be a double-edged sword. We know that when you combine a PD-1 antibody with CTLA-4 antibody, you have a substantially increased risk of fatal myocarditis, for example. And so safety is going to remain a primary endpoint in the next generation of studies. And infrequent but potentially devastating toxicities may not be detected in these small pilot trials and only become evident as we get into larger expanded studies, and so they will certainly remain a concern for some time to come. Well, what about stimulating HIV-specific immunity through the use of therapeutic vaccines? And there is now at least 2 decades of work that has been done in the attempt to develop therapeutic vaccines, and we still don't have a therapeutic vaccine for HIV. And one of the reasons we don't have one is illustrated in this study here. This was another AIDS clinical trials group study done several years ago and led by Chip Schooley using a Merck recombinant adenovirus vaccine that carried only HIV gag. And so here the top line is showing the placebo recipients, and the bottom line shows the vaccine recipients, and you can see they're mostly overlapping. Through the magic of biostatistics, actually, there was a small but modestly significant effect at the final time point in terms of the set point level of viremia, and overall, the viral set point was inversely correlated with the proliferative responses of CD4 cells to HIV gag and interferon gamma responses. Now, one important thing that happened in this study is that the study was supposed to have enrolled something like 120 participants. We got to participant 113, and it had taken us long enough to enroll the study that the vaccine was going to expire before we could complete the vaccination of patients 114, 15, and so on. So we were unable to finish enrollment. Now, why was the vaccine going to expire? By the time this study got done, Merck had already decided to abandon this vaccine in its development. And that has happened to every single therapeutic vaccine that's come forward to date, except for the few that are still most recently in study. And that has deprived us of the ability to learn from this result and to try to improve on the result, because nobody continues to build on the vaccine candidates. And every time there's a new therapeutic vaccine that's studied, we're essentially starting from square one, because with each vaccine, the people producing the vaccine think they have a different assay that monitors response best, so we can't say whether this vaccine was better than an earlier vaccine or not, because it's a totally different assay. And we always see roughly similar results, a modest effect on set point viremia. So we've learned that nearly all candidate vaccines have good immunogenicity in phase 1 and phase 2. This vaccine was terrific at inducing immune responses. We see encouraging results in a variety of non-human primate studies, but it's been very hard to replicate those in people. And there have been only very modest effects, half log typically, in post-vaccine control in the human trials, and no consensus on correlates of vaccine efficacy. That shouldn't be too surprising, because we don't have correlates of vaccine efficacy for a preventive vaccine yet. And there's been no systematic approach to building on the results of previous trials. So what we really need are to define the endpoints for our clinical trials and to have uniform endpoints from study to study, and to attempt to define correlates of activity in order to have a sustained focus on the most promising vaccine candidates, and then to be able to define long-term objectives and have clear go, no-go criteria so that we can push forward those vaccine candidates that are most promising and terminate earlier those that don't seem to be getting us where we want to be. There's been enormous interest recently in the potential use of broadly neutralizing antibodies to help control plasma viremia, either as long-acting therapies, and I won't speak about that because that's really the therapeutic use of antibodies as opposed to their use for remission or cure, or to harness the innate immune system to target the reservoir. So Pablo Tebas and Katie Barr, as well as Teo Chun did 2 separate studies simultaneously, jointly submitted them to the New England Journal of Medicine, who then said, you're going to have one paper, put them all together. And so that's how these got published side by side. They found exactly the same thing. They had patients who were on suppressive antiretroviral therapy. They gave them an infusion of antibody, stopped their antiretroviral therapy, continued the antibody, and not surprisingly, the patients rebounded. And the reason for the rebound is there was pre-existing drug-resistant virus that then emerged and continued to replicate in the presence of these antibodies to which the virus was not suppressive. And we've seen similar data from Michelle Nussenzweig's group and Marina Kasky at the Rockefeller. The big question is whether these antibodies actually have any effect on reservoir, which has not yet been demonstrated, and whether they have vaccinal effects, which was a suggestion from the Rockefeller studies that perhaps there's been some induction of better virus-specific neutralizing antibodies, and whether that could play some role in addressing the reservoir or in enhancing remission remains to be determined. What we have seen is really striking data from Dan Baruch using in monkeys now, and this is the only monkey data that I'll show you, using the TLR7 agonist I mentioned previously in combination with another broadly neutralizing antibody, PGT121. PGT121, like VRC01, is also a CD4 binding site directed antibody. And what you see here is in the monkeys that received the sham treatment, that so the monkeys were infected, put on suppressive antiretroviral therapy, and then after treatment were released from therapy. And here all the sham monkeys rebound. If they got the TLR7 agonist, they all rebound. If they got PGT121, there's some of the monkeys have a delayed rebound, and then with the PGT121 and TLR7 together, there are even more monkeys who have a delayed rebound or a blunted rebound. And so this is really quite intriguing. And some of these monkeys, when they then did passive transfer experiments, they couldn't demonstrate the persistence of any infectious virus in the monkeys that were aviremic. And so very intriguing results. And there are, as I mentioned, phase 1 trials of the TLR7 agonist ongoing in people, and phase 1 trials of PGT121. And so it won't be too long before the two can be combined in people to see whether this result can be replicated. So we've seen modest delays in time to rebound with the BNAbs that have been used individually to date. No demonstrated effect in humans on the viral reservoir, and that's because of the pre-existing resistant minority variants. And so we'll have to use these antibodies in combination or as genetically engineered bi- and tri-specific antibodies. So the questions that we need to address are, can these broadly neutralizing antibodies or the bifunctional antibodies that target virus through one arm and harness effector cells through another arm, redirect innate immunity against latently infected cells? They certainly do that in the laboratory. Whether that is an effect that is seen in infected people has yet to be determined. Whether in the human trials to date, the BNAbs are really targeting productively infected cells and destroying them through ADCC-like mechanisms, or are merely accelerating the clearance of accelerated immune-mediated clearance of viruses, not yet been determined, and whether these antibodies really are going to have a role in cure, or whether they're best thought of as potential long-acting antiviral drugs to maintain suppression in place of daily oral therapy is something that is now under active study. So let me say a word about the importance of correlates and surrogate markers for HIV cure. I've mentioned a few times that we don't really know what we're measuring and how best to measure the reservoir. And because there are numerous interventions that are being investigated as potential approaches, and because test of cure is ultimately going to require treatment interruption, we have to have some means of prioritizing which of these interventions we're going to put together in combination, and when have we actually observed enough of an effect that a treatment interruption is warranted as a test of cure. And so measuring the relative effects of candidate interventions on surrogate markers could guide improvements in these approaches. Then another important element in how we go about doing clinical trials for HIV cure or long-term remission is the need to do treatment interruption. I think despite some initial hesitancy, it's become clear that at least short-term treatment interruptions can be undertaken safely if done with appropriate monitoring. There's still some debate about whether we should just be looking for time to rebound or whether we can allow people to get to set point, and if so, just how high we can allow viremia to go before we intervene. The risks are those of primary infection syndrome. Our two patients developed a symptomatic primary infection when they rebounded because they had naive immune systems. There's more significantly the risk of transmission. If somebody is in a relationship with a partner who's uninfected and they've been on suppressive antiretroviral therapy, we know that's effective prevention. Now that they go off their therapy, if they rebound, they may then transmit virus, and so their partner has to be involved in the decision to embark in this interruption. There's the theoretical risk of reseeding the reservoir, and I'll show some data in a minute about that. And then the relative, so I've already talked about this issue of time to rebound versus set point. And what about doing interruptions in people who have received the placebo? We know what's going to happen. They're going to rebound. But you can't be sure that you're observing an effect, especially if the delays are modest in the initial trials, unless you have a comparison. But when we don't have a placebo, and many times we have a control group, but they're not getting a placebo, so the participants themselves know that they're not getting any effective therapy, it may be much harder to convince people to undergo the interruption. And then there's an ongoing debate about when treatment should be restarted in these trials. Interestingly, at a forum for collaborative HIV research discussion at the end of January, where the issue of treatment interruption came up, Jeff Murray, who's one of the people on the antiviral group in the antiviral branch of the FDA, said he was much less worried about treatment interruption than he was about the dangers of some of the drugs that we were proposing to give our patients. So that was an interesting perspective. So I mentioned this issue about reseeding the reservoir. Well, it may be that the reservoir is not so easily reseeded. John Lee in my group has looked at patients in ACTG trials who underwent treatment interruptions of various kinds and became post-treatment controllers and then went back on therapy. And if you look at people before and after the treatment interruption, you actually see that in, at least as far as measuring integrated DNA copies, you can see that there's really no net change. And if you did the ratio of post-interruption and pre-interruption, although there's a twofold variation, there's certainly not a huge reseeding of the reservoir that's occurring. And several other people presented very similar data at CROI just last month. So let me conclude with some unanswered questions. We have to determine what is the minimum clinically acceptable duration of drug-free remission. We talk loosely about remission, but nobody really defines what we mean. If you have to go on antiretroviral therapy, become suppressed, get a vaccine for 6 months, then get a PD-1 antibody and some other cytokine, IL-15 or IL-10 blockers, since Rafik is here, how long does the benefit have to be to justify a 1 to 2 year intervention to get there? And what proportion of individuals have to be successful in order to justify that approach? Is remission or cure sufficient to return the pro-inflammatory state to normal? And if so, will that eliminate or reduce the risk of non-AIDS events? That's still a hypothesis. Nobody has any evidence that that is in fact the case. How can we determine the effect of remission or cure on the risk of transmission? If somebody is only in remission and they became pregnant, would we not give that woman antiretroviral therapy to protect the fetus? Or would we consider that she still potentially has a risk of transmitting virus? That's almost an unanswerable question because it would be very hard to do the experiment ethically. And I think the really the billion-dollar question is, at what point can we equate sustained remission to cure? If somebody is in remission for 15 years, 20 years, 30 years, when can we finally say, yes, you're cured? And I don't think we're going to have an answer to that question for a very long time. So let me end here just by acknowledging a very large group of people who've helped with some of the studies I described, as well as shared some of their data with me. And I'll stop there and happy to answer questions.
Hi, Professor. Based on the experiment that mimics the Berlin patient transplantation, what would be the consequences of the absence of the CCR5 gene to the patient?
Yeah, so that's a great question. So it turns out that the 1% of people who lack CCR5 have no definable phenotype. There is some evidence that they may tolerate organ transplantation somewhat better. Of course, that's a rare event, as you might imagine. And there was some concern that they may be more likely to have progression of certain neurotropic viral infections, but that's never been proven. So by and large, there's no phenotype associated with it. They seem to be healthy. It was that observation that encouraged folks about 15 or 20 years ago to pursue the development of small molecule inhibitors for CCR5, and there is an approved drug, Maraviroc, which blocks CCR5, and that drug is generally pretty well tolerated. So knocking out CCR5 is not so bad. Knocking out CXCR4 is another problem. That's the other co-receptor.
Do T cells actually express toll receptor 7?
I couldn't answer that.
Do? I don't think they do.
No, they don't.
I think actually what has been shown, I believe, some of the in vitro work, is that essentially TLR7 is acting to produce interferon, and so you're getting local production of interferon, but at much lower levels and without the systemic side effects of giving interferon. So this is essentially a better tolerated approach to administration of interferon alpha.
Yep. Another question?
And do you know if the interruption of the therapy would enhance some of the chronic consequences on the HIV patients?
I'm sorry, can you repeat the question?
Do you know if the interrupting the therapy would enhance get worse in the consequences?
So what we do know is if you do repeated interruptions, that people will have serious effects. And so the SMART study, which did intermittent therapy, where people were put on therapy and then they were taken off treatment when their CD4s went above 350, and then they were taken off treatment again until their CD4s declined. The people on the intermittent therapy arm did much worse. They had more AIDS progression, they had more heart attacks and things like that. But doing a single treatment interruption of 16 weeks doesn't seem to have any consequences. You can show increased inflammatory markers during that time, but whether that brief period of increased inflammation has long-term health consequences seems less likely. Rafiq?
One comment and one question. So my comment, you probably know that we've done also a lot of work with the Sangamo product.
Yes.
And in heart-rate subjects, we've seen up to a 1 to 3 log decay in integrated DNA and in tilde values, at least a log. And one of the major correlates of that decreased reservoir was the fact that we were getting non-cognate help to restore CD8 responses. So I think one potential mechanism of action of those infused CD4 T cells is to provide non-cognate help to HIV-specific CD8 T cells.
Right.
And that's one way of going about it in an upcoming trial. My question to you, which I think I know my answer, but I would like to know your answer, is whether do you think, and I hope you're going to say no, that it's one treatment, and I mean, are you going to get one cure for everyone, or you think that's going to be much more personalized, and how would you go about defining who gets what kind of treatment?
Yeah, that's a great question. So in case people didn't hear, the question was going to be, is it going to be one cure for everybody, or are we going to have to individualize our approaches to cure? I think it's almost certain that we will have to individualize. It will... First of all, the approach will be a combination of approaches. It won't be a single intervention, and it's likely that until we see what works, we won't know how we're going to categorize patients and decide who gets which approach. Michael.
So Dan, you talked about both vaccines and cure. So if you had to bet, what do you think we'll have first? A scalable cure strategy or strategies, or an immunization, passive or active, that'll prevent acquisition of infection?
Well, I would say a cure, because actually, so far, if you did the math, we've done much better with the CCR5 deleted stem cell transplantation as a percentage of the N compared to the protective effects of vaccines. But you said scalable.
I did say scalable.
You did say scalable, yeah, well.
Then I have a question to you, but not scientific. You've been working with a lot of those trials, and how easy it is for you to convince patients to do ATI, to do interruption of the treatment?
It's not so difficult. I think it depends on the patient. It depends a lot on their clinician. I think that the biggest challenge is helping people understand why the interruption is being done, and that it's only a temporary interruption. There are lots of people who'd like to come off therapy altogether. And then so part of the challenge of doing these studies is saying, we'd like you to come off therapy, but only for a certain period. And we really want you to go back on treatment afterwards. And sometimes it's hard to get people back on treatment, and that's not a good idea. So we're now doing a study that John Lee and Davey Smith are leading, trying to determine whether we can find any correlates of time to rebound. And in that study, which is attempting to enroll something like 80 participants or so, there's no intervention. It's just people come in, we do a leukapheresis, we take them off therapy, they get monitored twice a week, and then they go back on therapy. And we've got that study about half enrolled already. So it's going much faster than we thought.
Just a comment.
Sure.
We participated in this SMART study,
Yeah.
and the START study also. And I guess the problem is not the patient. The problem is how to approve ethically the proposal of, again, suggesting the STI.
Yeah, no, I did that.
That's the point.
Right. So that's a great point, and that's why I mentioned that the FDA is not so worried about the interruption. They're more worried about using pembrolizumab or giving romidepsin to people. Yeah. But local human subjects boards are concerned. I think it's a process of education. I think it's important that both the community and the local ethics authorities have an opportunity to understand the context in which these studies are being proposed, why they're being proposed, and that the field as a whole feels that this is an important experiment to be done, and that it can be done safely with minimal risk to the participants.
Any more questions? Thank you.
Okay. Thank you.
Thank you.
Yeah. Okay.